everybody. My name is Dr. Roma Chilengi. I work for the Center for Infectious Disease Research in Zambia. In short, we call it CIDRZ. I'm pleased to talk to you about this program. So I graduated from the University of Zambia as a medical doctor with a medical degree. And having worked a few years, I moved into a research career full-time. I have had the privilege to be trained at master's level at the London University in the London School of Hygiene and Tropical Medicine, where I studied epidemiology. And beyond that, I've studied vaccine development and clinical research, and spent my postgraduate fellowship time in Belgium. I'm happy to talk to you briefly about the work that I do, which is biomedical research. This is a very broad field of medical discipline, but a part that is of relevance to this conversation has to do with what we call clinical research. And in clinical research, it's basically a field of study when we are trying to answer specific questions, whether it's about how particular infections cause disease, or how the human body responds to that disease, or indeed how we develop specific drugs or vaccines or diagnostics, as it were, to help the doctors in practice deal with those particular diseases. This process is very well regulated, and it involves quite a number of steps. The primary thing that I would like to emphasize is that before any research is done legitimately, we describe that work in a document we call a study protocol. And by describing a study protocol, we include in there everything that relates to that specific work that will address a specific question, meaning that there are approvals that must be granted, firstly to the team of researchers who are proposing to do that work, but then to the nature of the investigation they are trying to do. And there are multiple levels at which those approvals and reviews are done. The primary review is done by a body of experts we call the ethics committee. And in our case, we usually go to the University of Zambia Biomedical Research Ethics. And once the ethics committee gives specific approval to a study protocol, we then must go to the Zambia Medicines Regulatory Authority. In short, we call it ZAMRA. And ZAMRA is the organization that is empowered to make sure that medicinal products and interventions that are used on the people in a given country, like Zambia in this case, are reviewed and approved before they can be given to the public. And beyond ZAMRA, that is after we obtain the ethics review approval, the ZAMRA review approval, that's when we now go to the Ministry of Health, we call it the National Health Research Authority, who give the final review and authorization to undertake research. Well then, having introduced the program, I would like you to hear for yourselves from the Ministry of Health. Now, biomedical research and all the work that we do really occurs at the auspices of the Ministry of Health. And it is important that we get a word from the Ministry of Health.
Thank you very much. You are all welcome. I'm Dr. Kennedy Marama, Permanent Secretary in charge of Technical Services in the Ministry of Health. I'm a public health specialist with 24 years experience working in the public sector at various levels, starting from the community, district, provincial, and now national levels. For us in Zambia, when we look at the socio-economic development of this country, we are guided by the overarching government agenda. And this is contained in the National Development Plan. So for now, we are saying we need to accelerate efforts towards development without leaving no one behind, so that we attain the Vision 2030. Vision 2030 for Zambia is to make our country a prosperous middle-income country by 2030. For the health sector, We are guided by the National Health Strategic Plan for now, 2017-2021. Our role and the key mandate is to ensure that we make our people healthy and productive, because that is what is going to contribute to the socio-economic development of our country. The key question which comes is, how can the health sector then catalyze development and ensure that our people are healthy and productive? One of the key issues comes to research and innovation. Research and innovation is a key investment for Zambia. And this is the reason now Zambia has what we call the National Health Priorities for Research. And also we have a National Health Research Agenda for this country. This agenda articulates or stipulates priority areas in which researchers should focus into, because one of the challenges with research is that if it's not well coordinated, regulated, and the people are doing research well monitored, you may end up with research topics or subjects being investigated which are not speaking to the agenda of the country. So we are happy as Zambia, as government, as Ministry of Health, all the research topics or issues in the health sector which are being explored are in line now with the government priorities. So we need to ensure that whoever wants to do research, the health sector is on top of things and we guide them. And this is very important to our people, because if this is not done, then we have people who are coming pretending to be doing research and it's not for the benefit of our people. So to our people in Zambia, be rest assured once again that government, through Ministry of Health, will always ensure that whatever research is being done in Zambia, it is in line with the standards which are laid down. Were you worried of being a participant in any research authorized by the Ministry of Health? I'm sure after listening to this, you are game to go. Wish you the best. Thank you very much.
That's the voice from the Ministry of Health. Thank you so much, and we are very, very grateful for the support and the umbrella we get from our mother body in the Ministry of Health. I would now like to bring your attention and listen to Professor Godfrey Biemba. Professor Biemba is the Chief Executive at the National Health Research Authority. This is the organ that also oversees and gives us authorization to do research. Professor Biemba, over to you.
Hello, my name is Dr. Godfrey Biemba. I'm the Director and CEO for the National Health Research Authority. I'm a medical doctor by profession, but I'm a researcher also. I've been doing research for the last over 32 years now. I hold a bachelor's degree in science, human biology, that's from the University of Zambia. I also hold what we call Bachelor of Medicine and Bachelor of Surgery from the University of Zambia as well. And I hold a master's degree in public health in developing countries from the London School of Hygiene and Tropical Medicine. So the National Health Research Authority is a statutory body that was formed way back in 2013 by an Act of Parliament, Act number 2 of 2013. The National Health Research Authority has a mandate to regulate research, to promote research, to coordinate research, and to build capacity among researchers, research institutions, and research ethics committees. It has also got a mandate to ensure that all research that is conducted in this country, in Zambia, is translated into policies and programs. We register researchers, research institutions, as well as research ethics committees. Now, I must say that research ethics committees are responsible for ensuring that research participants are protected from any harm when they participate in research. The process is such that when anyone wants to conduct research in this country, they will have to apply to a research ethics committee. And once we are satisfied that the research ethics committee did a good job and we are satisfied that there are no issues of security concerns to the country, then we approve that study and the study can proceed. So that is basically the role of the National Health Research Authority. 1, to regulate; 2, to promote research; 3, to build capacity; 4, to coordinate research; and 5, to ensure that research results are translated into action and policies; and lastly, to advise government. So the National Health Research Authority, as I indicated earlier, is the main regulator of all health-related research in Zambia. But you see, it doesn't work on its own. It works with other regulatory bodies. And specifically, I'll mention the Zambia Medicines Regulatory Authority. I'll mention the National Biosafety Agency or Authority, NBA. Let me say something about CIDRZ. CIDRZ is an institution that has been in the Zambian market for a long time in terms of research and has done a lot of cutting-edge research, especially in HIV. And we have worked collaboratively with CIDRZ for a long time. And there's a lot of research that CIDRZ has conducted that has ended up in policies and programs that have changed the lives of the Zambian people. So we recommend CIDRZ as a force to reckon with as far as research is concerned. So viewers, would-be participants, with all the information that I've given you, with all the assurances that I've given you, I hope you feel free and comfortable to participate in clinical trials. Clinical trials are extremely important because all the drugs that we're using now come from clinical trials. When you see people being treated for malaria, when you see people being treated for cancer, when you see people being treated for HIV, all these are products of clinical trials. So I want to encourage you and assure you that you are protected. Feel free to get involved in clinical trials. Thank you.
Thank you, Professor Biemba, for that discourse on the National Research Authority and its role that it provides and serves us as we undertake research. I would like you to hear for yourselves from Dr. Sodi Munsaka, who is the chairperson from the University of Zambia Biomedical Research Ethics Committee.
Hi, my name is Dr. Sodi Munsaka. I'm the chairperson of the University of Zambia Biomedical Research Ethics Committee, which is an institutional review board for the University of Zambia. In terms of education, I graduated from the University of Zambia with a degree in microbiology and then went on to the US state of Hawaii, where I did a master's as well as a PhD in biomedical sciences with specialization in tropical infectious diseases. Thereafter, I decided to come back and join the University of Zambia to teach as well as conduct research. It is through that research background that I was appointed as chair of the University of Zambia Biomedical Research Ethics Committee.
Thank you so much. That was Dr. Munsaka giving a view of the role of the University of Zambia Biomedical Research Ethics Committee. I would now like you to listen to Mrs. Bernice Mwale, who is the Chief Executive of the Zambia Medicines Regulatory Authority, in short, ZAMRA, who will give her perspective on the role that ZAMRA plays in the process of clinical research.
Hello, my name is Bernice Mwale and I'm the Director General of Zambia Medicines Regulatory Authority. I'm a qualified pharmacist. I studied pharmacy in Scotland and I've been a pharmacist for a long time. And in relation to clinical trials, I have a postgraduate qualification from the University of London, and I'm currently overseeing all the functions of the regulatory authority. The Zambia Medicines Regulatory Authority is a medicine agency that has been created by an act of parliament. the Medicines and Allied Substances Act number 3 of 2013 to ensure that all products that are being made available to the Zambian public are safe and of good quality and efficacious. So one of the functions of the Zambia Medicines Regulatory Authority is to regulate and monitor the conduct of clinical trials that involve medicines. And before that, we also inspect the sites where the clinical trial is supposed to be conducted. And in that way, we look at issues like the number of study personnel that are there, the infrastructure, and so many things, so that we are satisfied that the people that are conducting that clinical trials are qualified and so forth. In addition, ZAMRA also gives permits for importation of the investigational medicinal products that are used in studies. So the institution conducting clinical trials cannot bring in the products without permission from ZAMRA. The Center for Infectious Disease Research in Zambia, in short CIDRZ, is an important institution that conduct various studies or research, and the data that is generated also assists ZAMRA in terms of either registering a product, in terms of the indications. You know, sometimes something like an example I can remember off cuff is where a medicine is registered for certain indications, but after studies you can add additional indications, say in children, or it can be used in children and scaled up to adults. So it's important that research is done in Zambia, and also it provides ZAMRA an oversight for research that is done in country, rather than the research that is done elsewhere. And also the data that is generated is particular to the Zambian population, and that assists the end users, because the data that is generated is really specific to the Zambian population. So it's important that we have institutions like CIDRZ to conduct research within Zambia. And so to the participants that participate in these studies, I just wish to assure them that there's regulatory oversight from all angles. We have a part to play as ZAMRA, but there are other institutions like the National Health Research that also looks at ethical part of conducting clinical trials. So you shouldn't worry, because you're really helping us to have data that is based on the Zambian population.
Thank you, Mrs. Mwale. That's a view from ZAMRA. At this point, it would be important to actually show the viewers a real research site and real clinical research going on in the field, so that they get an appreciation of what we do when we do clinical studies. So welcome to George Research Site. This is one of the clinics in which we are doing a number of vaccine development trials. We are particularly doing clinical trials that are evaluating vaccines that are in development against diarrhea, vaccines that will protect children against diarrhea. At this point, I'm standing right in the middle of the clinic, and in my back, some of the research participants, and Some of our staff are picking up documentation for the studies. But generally, this is a typical research clinic where we recruit participants and we get their consent, and then we examine them and we proceed with clinical trial design.
Hello, I'm Dr. Suko. I'm one of the research clinicians here at the CIDRZ Clinic research site. At the moment, we're doing clinical trials on rotavirus. So we're testing a new vaccination. So as you can see, today we're doing enrollment. So we have a lot of mothers here at the clinic. So on a day like this, it looks a bit similar to how you'd find it in a clinic where there are a lot of mothers waiting to be attended to. So we're also equally very busy. So we see the babies. Normally we get healthy babies to be part of the clinical trial. And then we enroll them. And then we just make sure that they're okay. And yes, then we follow them up until the end of the trial. So the main difference between doing clinical research and clinical work is most of the time when the participant comes to the clinic, it's not usual that they have to sign a document. So here they normally have to give what we call informed consent. So that's permission for us to do procedures with them and to also to take part in the study. So that's the main big difference. It's also fast-paced, just like in the hospital. We get a lot of mothers coming in, and then we also screen them to see that they're healthy. So usually in the hospital, I'll be taking care of babies that are sick, but here I'm seeing healthy babies. Then the other things that we normally do, because it's a vaccine trial, we'll also get samples, so blood samples. Sometimes we also get breast milk, and sometimes even stew. So it depends on what we want to find out about how the vaccine is working. So that's one of the big differences between clinical work and here and doing research.
My name is Dumisani. I'm the farm tech for the research site here at George. And this room that you see here is a pharmacy room where we keep our vaccines. These vaccines that we are doing here are on trial. So we stock the vaccines in this fridge. This is a 2 to 8 fridge, which is monitored by a hobo logger. In case of any temperature excursion, it rings to inform us of what has happened. So we keep the vaccines stored at 2 to 8 degrees. And these are the vaccines that we do give to our children that come.
Hi again. Thanks, Dumisani, for explaining. I think it's important for our viewers to be aware that we look after study vaccines very, very carefully. The vaccines we are working with must be kept between 2 and 8 degrees. So the freezers that you see, like this is a special vaccine fridge which monitors the temperature, and we have double systems for monitoring the temperature. And the system we have here is such that should power fail, there is backup UPS and the backup power systems that keep the fridge running for at least 6 hours. And should the temperature come out of range, immediately either himself, myself, and a few other people will receive messages on our phone to say something is wrong with the vaccine fridge. Wherever we are, we have to come immediately and rescue the study vaccine. And this is done in order that, because these vaccines are under investigation, so we want to be able to evaluate them so that if the vaccine works or doesn't work, we assess it as objectively as possible. You can imagine sometimes you might decide that the vaccine doesn't work when in fact it went out of the storage temperature. So in terms of the way we care for vaccines, it's extremely important in clinical trials to maintain the temperature. And these are not for public use, they are for research purposes alone. And that's why he was showing you the accountability and how we have to count every vial. And actually the sponsors send their monitors to check that only people who are enrolled in the study are receiving these vaccines, and we are not even at liberty to throw away used vials. Those have to be counted one by one and are handed back to the manufacturers. So this is the mini lab here at the research clinic. You just noticed Sister Margaret passing on a blood sample to Sekai. Miss Sekai is one of our biomedical scientists in the lab. So what she does basically is receive the blood as is collected, and she will do her documentation and then put the sample in this little machine here. We call it a centrifuge. So the centrifuge, what it does, it spins down the blood at a very high speed. And what that does is it separates the cellular components of the blood from the liquid part of the blood. So the liquid part of the blood, which we are interested in, it will be called the serum. If you get the blood from the body and you take a long time, you then interfere with the levels of those immune responses. So what this process helps us with, it separates the blood, and in that state, under temperature controlled condition, we then move the blood sample and take it to the main lab. In the main lab, that's where we do those detailed immunology stuff. But in this lab, we have to just prepare the samples so that when they get to the main laboratory, which we will show you shortly, you then can analyze your samples to get the results that you are looking for. So this mini lab really helps us prepare the samples. I want to show you the transportation mechanism. We use these, they are special cool boxes. When the samples are ready, they are put inside these cooler boxes. Inside these cooler boxes, as you can see, there is a gadget there, and it has this cigarette lighter plug. So we plug them into the vehicle cigarette lighter for power, and then that power maintains the temperature in here during transportation, so that the samples arrive in the lab at the correct condition. So David, incidentally today, I would like you to be able to walk our viewers through the procedures that our samples, which we collect from the participants, go through. We would like them to understand why we collect those samples, and I think you will be able to show the viewers through the various benches what we do with these tests. I would really appreciate for you to give a bit of overview about the lab. What do you do here at the lab?
Thank you, Dr. Chilengi, for looking into this overview. In brief, when the sample gets to the lab, the sample is assessed for its quality standards, whether it meets all the requirements that allows the scientist to test the sample. Once that is done, then the sample goes through information capturing to make sure that the information that is captured relays exactly what came from the clinic or from the study, and that it's the accurate information that is sent back to the clinic. There is no participant or patient information that needs to get into the system if it's not accurate. And usually what we do, we get back to the clinical team or to the clinic and say, give us additional information for this participant so that we capture what is accurate. And once that information is captured, as you will see during the process, the information moves electronically. What we understand at the lab here is that even if you, the participant, don't come to the lab, but your sample is you, and we treat every sample as a face behind it, as an individual. So we are very cognizant of that, and we take care of every sample individually, and it's treated with respect.
Thank you, David. Well, I hope today you will allow us to be able to show the viewers and indeed some of those participants
Yes.
what we do with the samples. I hope that you will invite us into your lab and be able to show us the various steps that the samples are taken through.
We're very glad to let you look into our lab and have a clear understanding of when you give us your sample, what your sample goes through and how we process and test your samples from our laboratory and its journey to other international laboratories that are specialized in the kind of testings that we might not have locally or nationally.
Well then, I think there must be a lot of excitement and anticipation out there. Why don't we get right into it so that
Fantastic.
we begin to show these processes.
Well,
thank you, David.
Please.
Yeah.
Thank you.
Hello, my name is Jacinta Sandra Mieva. I'm a technical lab assistant. Basically what we do here is receive samples from the various clinics that we service. The main job for this department is to ensure that all the samples that we receive are verified and checked for clarity and ensure that the demographics are matching on the specimens as well as on the requisition forms. So we have three main stations: the sorting station, where we receive samples and sort them according to the clinics and the tests that have been requested. From there onwards, we take the samples to the data entry section, where samples are entered into our LMIS, which is the lens that we capture information and verify that the details in the system as well as what is appearing on the requisition form are similar. If there are any discrepancies, then we withhold that sample for further verification. If it can't be verified, then it is rejected and the clinic is informed. From there onwards, the samples are entered into the LMIS, where we generate barcodes which are stuck on the samples at the sample labeling station. So the job of an assistant is to ensure that what they have, the requisitions, the specimens, as well as the samples, are in accordance with what has been entered. If there is any discrepancy, then as well at that session, the samples are verified and checked for clarity. If they can't be controlled, then they are rejected and the clinics are informed using our laboratory tool that we use, which is the specimen problem log.
Welcome to chemistry department. I'm Winga Louis, as he has explained. I'm a biomedical technologist on this department. So our department is divided in three sections. We have the clinical chemistry department here, where we use the Cobas C111, the 2AU480s, and on the serology department, we have the access to immunoassay, as well as the ELISA equipment at the far end, that side. So on the clinical chemistry, the main tests that we do are liver function tests, kidney function tests, and electrolytes. So these tests give us a picture of how well body organs are operating.
This is the hematology department, which looks at blood and its components. Blood as it is, it has a different number of cells, which includes white blood cells, red blood cells, and platelets, which are suspended in a liquid part, which is plasma. And at this bench, this is where we do the testing of blood cells. We look at each and every different type of blood cells. And this here we do the testing. We'll go into the machine will do the mixing so that the cells are evenly suspended, and then to aspirate and do the testing and the counting at the same time. And the results will go to the computer there. So what we do as a technologist, we look at the results. If you are satisfied that the results are okay, then you validate, meaning that you have accepted the results. Once you validate the results, again you come to the laboratory information system, that is to go into the computer system. You still validate, and then the results will print out. If you are satisfied that the result is okay, once the validation is done, then it will print out.
My name is Larry Daka, and welcome to flow cytometry bench. So at this bench, what we are testing is blood. So we receive whole blood in the vacuum containers. So we receive the blood in these containers. So these containers, we load them in the cassettes. And the cassettes we load in the instrument. So this is how we load. So the cassette, I'm loading in the blood. Now when we load, we mix, and then we load in the instrument. So this instrument is going to analyze. And when it finishes analyzing, it's going to transfer into our information system. Thank you very much.
My name is Kaunda Kaunda. I'm the manager for the CIDRZ TB laboratory. In this lab, we're in the reception laboratory. This is where we receive all specimen or samples that come from patients. Mostly the samples we receive here are sputum samples, which is from the chest, not saliva, but sputum samples. And when they come in here, they come in biohazard bags with requisition forms. When they are received for sputum samples, especially for culture, we always maintain a cold chain, so the samples are kept in a refrigerator while they are being documented. Reception is done using a computer, which is called the LIMS, so that we can keep track of your samples throughout their lifespan at the CIDRZ Central Laboratory. And the tests that we do for the samples from sputum is we have smear microscopy that looks for the bacteria that causes TB. It's called Mycobacterium tuberculosis. Then we also do GeneXpert, which is a more sensitive test. It uses molecular methods. It also determines whether you have TB bacteria in your sputum and whether that bacteria is rifampicin resistant, which is one of the drugs that is used to treat TB. Then we also do culture, which is where we get your sputum and grow it up so that even if you had few bacteria, we can grow them enough to know that you actually had bacteria and be able to do some other tests beyond the simple tests like smear microscopy and GeneXpert that we spoke about. To my left here, we have our biosafety cabinet, because TB, as you know, is because when you cough out bacteria, so it's airborne. And for safety purposes, even when we handle the samples, we use a biosafety cabinet that will protect us as well as the sample.
Welcome to CIDRZ Central Lab. You are in the specimen repository area. I'm Rabson Mwanza, a laboratory archivist, and my colleague is Paul Pandala. I think he's going to walk you through what goes on into this specimen repository room.
This is our specimen repository room. We receive specimens of different types, blood, plasmas, serum, We also do PBMCs storage here, vaginal swabs, and mucosal specimen. These are the freezers. We have got different freezers with different temperature for storage according to what specimen temperature requirement is. As for this one, this is the -80 freezers for the long-term storage of specimens which are stored in here. We do monitor of temperatures twice a day at 8 hours and at 16 hours. We monitor temperatures on all freezers and the rooms in here 24 hours. But we take readings manually twice a day at 08 hours and at 16 hours. We also have the online monitoring system that will send messages, emails in case of any temperature deviation. This room has got its own backup generator just specifically for this room. In case our solar and the ZESCO grid is not working, the bigger genset also is at 40. There's a specific genset just to cater this room. This room is not supposed to go without power at any cost. So we are all alert 24 hours to monitor temperature in here. Down inside. These are the freezers that for my own protection, I cannot touch this freezer with these gloves. I might have frostburns, so we use the hygienical gloves. These are -80 freezers. It has got the range of 65 to 85. That's the range. So this one is 78, which means it's within the required temperature range, which is 65 to 85. With different freezer temperature range for different storage requirements. There are some specimens which are required to be stored for a longer period of time, which are stored at -80. For these ones, we have got -30 freezers, which are for short-term storage. The range for these freezers is -15 to 30. These are the short storage freezers. In conclusion, the definition for the short storage and long storage is that for these ones which are stored at -30, they are short. They can be stored for a year or 6 months. These are the samples that are going for testing, not for storage. For the ones for the -80s that we have seen for, those are for the long-term storage or indefinite storage. They can stay forever in the -80 until test is required for the researchers. When samples are processed from the reception for archiving, they come through to the specimen repository for archive. We have got a system that we use to archive the samples, which is the Laboratory Data Management System managed by FISTRI Frontier Science of USA. When the samples are received here, they are entered in LDMS, which is the Laboratory Data Management System. This system will allocate the position for the samples where to be stored. So from here, when you are logged in the sample, we just don't go in the freezer and put them anyhow. There is a system that we follow to when you enter them in the system, it will generate a box map which will tell you where to go and put the actual aliquot, in which freezer and at what position, so that in the next 5, 10, 20 years you want to look for that specific sample. Someone will just log into the system and able to fish the sample out without any problems.
Okay. So we are very grateful to our lab teams and all the work they do to support our research studies. At this point, we need to move and shift perspectives and hear from our community team. Let's hear what they have to say about research and CIDRZ.
Hi, my name's Agnes Chitoshi. I'm a community liaison officer with CIDRZ.
Hello, my name is Azimba Moses, a research assistant with CIDRZ.
Hello, my name is Jamia Puri. I'm also a research assistant working with CIDRZ.
So we wanted to share with you a bit of what the community team does in CIDRZ. Our primary role really is to reach out to the community. We go into the community to try and engage the community for the various studies that we're carrying out as CIDRZ.
Okay, so essentially our role is to work with the community, and I would like to say I'm very grateful to the different stakeholders like the CAB and also the NHCs that we get to work with every day at our facilities here as CIDRZ. I would like to thank them for the work that they do in helping us retain mothers and sensitizing the community at large as we work with them. I'm also very thankful to the community. Even as we go out to work with them, essentially our job in the first place is to make sure that these participants are retained in our research projects, and we are thankful for the community because we work with them on a larger scale. These people get to welcome us into their homes.
We want to appreciate the communities for the support that they render when we do these research studies. As much as we have a lot of myths and misconception, we want to thank the community that slowly but surely research is sinking into the community, and we are receiving a lot of support from the community. Basically, for research information that you might require to have, you can visit our sites, that is George Clinic, Matero First Level Hospital, and then you can visit also Kalingalinga Clinic or Kamwala Clinic. These are sites where we are mainly found.
Thank you so much. At this point, it will be important to also listen to one of our former research participants who has gladly agreed to give his testimony about participation in a CIDRZ research.
Zina yanga nine James Bulaya. Umozi w'abantu banateknako party mu CIDRZ study. Beina yamba study banachite explain concept no bwino. Then I joined, and then from there I've seen the benefits, and I'm looking forward to the good results that would benefit the general public and the community.
Wow, this has been quite a journey. I hope that the viewers have now a sense of what clinical research involves and how we do that research. We hope that this video communicates some important information about how we do research, and that it encourages would-be participants to participate in clinical studies. Thank you so much to everybody who've made this possible.